This is the first of the genetic series, Human Genome and Hereditary, Module 1, Lecture 1. I would just like to mention that this first lecture is probably the longest in the genetic series. It's also the most basic. This is meant for those that do not have a master's degree in molecular biology or didn't study quite a bit of that during their undergraduate, because this may seem quite simplistic to you. But again, this is not something you probably have to spend a lot of time on. So moving forward, chromosomes were first identified in 1959, and humans have 46 chromosomes, or 23 pairs. The genes are located on these chromosomes. Normally we have 46 chromosomes, 2 of each pair. Girls have 2 X's, and boys have an X and a Y. The size of the chromosome doesn't tell us how many genes are on them. For instance, the chromosome with the largest amount of genetic material is 1, followed by 2, followed by 3, followed by 19. Approximately 60% of our chromosomes have just filler or non-useful genes in them. In a male, the difference between male and female is that there is a Y chromosome, and the Y you can see is smaller and primarily has information regarding determination of the male sex. If I were to look at 100 males, they would probably look like this, and 100 females would look like this, would have 2 X's, but there are many different genes. So these chromosomes are really like a scaffold that hold the genetic information in very tightly wound. We have 26 letters in our alphabet. DNA has 4, adenine, thymine, guanine, and cytosine, and it's a combination of these letters that are a recipe that tell the body how to make proteins. And proteins represent cell membranes, enzymes, pretty much everything to construct the human body. If you think of a chromosome like an encyclopedia, think of a page like a story. Or I like to think of a chromosome like a recipe book. And a page is like a recipe that tells the body how to make something. Now, there are a number of different problems that can occur. One is a missense mutation, where if the sentence is supposed to say the car was red, if the word red is changed to hat, or even the letter E is changed, it changes what this means, and it doesn't really mean anything. Nonsense is there's an early stop codon, so instead of it saying the car was red, it just says the car. Insertion of either a whole word or a letter can change the meaning, as well as a deletion of either an entire word or a letter can change the meaning, therefore what the end product ends up being. DNA structure, again, we have our 4 base pairs, adenine, thymine, guanine, and cytosine, and the purines are adenine and guanine, and the pyrimidines are cytosine and thymine, and the AT have double bonds, and GC have triple bonds. Gene-rich areas have a lot more GC regions to them. The DNA is held in a double helix, 5' to 3' and 3' to 5', and where there is a thymine, it attaches to the adenine, a guanine, cytosine. And these 2 strands together are known as the chromosome. The nucleosome further helps package the DNA into smaller pieces. 140 base pairs of DNA wound around a histone is known as the component of a nucleosome. And this helps, so we just unwind, if we want to transcribe a certain gene, unwind a small area, open it up, and then make a copy. Next, we're going to talk about cell division. Mitosis is somatic division. This is how the body grows. A single cell divides into 2 identical cells. Meiosis only occurs in the gametes. Instead of dividing the chromosomes and then having 1 division, there's actually 2 divisions, and you go from 2N to 1N. There's also a special thing that occurs only in meiosis, which is extra crossing over between the chromatids, and this helps to increase variability in mixing of the DNA. So here we have mitosis. We have, usually in the interphase nucleus, you don't see the nucleus, and the chromosomes are very stretched out. We then start seeing the chromosomes getting, we can start visualizing them by prophase. By the end of prophase, we see 2 chromatids. In prometaphase, you start seeing the spindles. We then, in metaphase, they line up with the spindles. In anaphase, the chromatids start separating. And here, in telophase, we have 2 separate cells with the exact amount of genetic information as the initial cell had. So from 2 cells, we have 1 cell. Replication occurs during the S phase. This is when we copy our new DNA. And in replication, the double helix separates and new bases come in. So where it separates, if this is an A T comes in. If this is a G, a C comes in, so on and so forth. And it results in 2 copies of the DNA. The enzyme used for this is DNA polymerase. G2, this is actually a really important step. I like to think of it as a double, like the discount double check. We make sure that all the base pairs put in are correct. This is a very important stage to make sure no mistakes were made when copying the DNA. And this is how mutations occur. And again, the body, we usually fix about 99.99%, but that 0.01% is what can result in cells overgrowth, cancer, and other kinds of things, which we're going to talk about, in future lectures. So here we're going to watch a video on mitosis.
During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the 2 sets of chromosomes. Mitosis is now complete. After mitosis, 2 new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of 3 stages called G1, S, and G2. G1, or gap1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands, called microtubules, appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, Prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
Next, we're going to talk about Mendel's laws. Gregor Mendel was a botanist who did research on pea plants. He's actually known as the father of genetics. And he actually proposed 3 laws in how cells divide. And how gametes are formed. The first is during the production of gametes, 2 factors segregate so that the offspring inherits one from each parent. The second, law of chance, determines which of a particular trait is inherited from a parent to offspring. And finally, dominant inheritance is when one trait dominates over another. Recessive, both traits are equal. 2 members of a gene pair alleles segregate separately from each other to form gametes. Half the gametes carry one allele, while the other half carry the other allele. So here, if the parental genome was big D, only the gametes would only get big D, or little d, they would only get little d, but the F1 progeny would have one copy of each. The second law is independent assortment. The distribution of one allele into the gametes does not affect the distribution of the other. Therefore, it should be random contributing. So here we have allele A and allele B. The father will give, here's big A, big B, little b. You can give a big A and a big B, a big A, a little b, a little a, big B, and a little a or little b. And because they're independent, which means they're probably on different chromosomes or far away on one chromosome. Now we're going to talk about meiosis. The purpose of meiosis is a reduction in the number of chromosomes from diploid to haploid. This is essential in the formation of gametes. Segregation of alleles in meiosis 1 or meiosis 2, according to Mendel's first law. Shuffling of genetic materials are random, assorting of homologs in accordance to the second law. In addition, there's additional shuffling by crossing over between the two chromatids. This will increase genetic variation. So in meiosis, as I stated earlier, there's actually two divisions. So our chromosomes divide, but they go through two divisions, and at the end, it's one N. Whereas in mitosis, you start out with your chromosomes, you duplicate them, you divide in half, and you have the same at the end. So there's an extra really important phase in meiosis. It's not present in mitosis, which is in prophase 1. So leptotene. So prophase, like meiosis 1 is, meiosis 2 is basically mitosis. Meiosis 1 is when the extra things happen. So leptotene is when the chromosomes become visible and the sister chromatids begin to align. In zygotene, there's homologous chromosomes align and the synaptolemmal complex develops. So in zygotene, this is when synapsis occurs. Pachytene, synapsis is completed, and diplotene, this synaptolemmal complex breaks down. So these are all subphases within prophase 1 in meiosis 1. Diakinesis, now the chromosomes are maximally condensed. So here you can see here's dad's copy of this chromosome, mom's copy, and during synapsis, here the dad's copy has a little bit of mom's on it. During prophase of meiosis 1, the sister chromatodes, like I said, cross over to allow increase in recombination. And here we have prophase 1, metaphase, they line up on the spindles, anaphase, and telophase, we get two separate cells. In meiosis 2, as I said, the second meiotic division is really mitosis, except for that there's no cell division in between meiosis 1 and meiosis 2. That's the major, difference between meiosis 2 and mitosis. So here we have just another example of how this recombination can change the gamete. Spermatogenesis begins during puberty. It initiates in the seminiferous tubules, and these tubules are lined with spermatogonia. The process of meiosis and spermatogenesis takes 64 days, and one spermatogonia results in 4 sperm. Oogenesis, in contrast, begins during prenatal development. The oocytes migrate to the proper area and begin meiosis prior to birth. Several million oocytes are present at birth, but only 400 are ever ovulated. And at ovulation, what happens is the mature ovum gets most of the cytoplasm, and the polar bodies don't get much. So with one primary oocyte, we only get one mature ovum. Completion of meiosis does not occur until after fertilization. Fertilization takes place in the fallopian tubes. Once the sperm enters the egg, meiosis is completed. And then the sperm nucleus enters with the ovum nucleus, and fusion occurs. Now we're going to watch a video on meiosis.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, meiosis 1, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis 2, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the twins are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the 2 poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into 2 daughter cells. Each of these 2 cells now undergo meiosis 2. Meiosis 2 is similar to mitosis. The first stage of meiosis 2 is prophase 2. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells have only 1 copy of each homologous chromosome. So in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the 2 haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Next, we're going to talk about genes. Genes code for proteins. The process of using DNA as a recipe is known as transcription. The DNA unwinds a certain region and copies the DNA into RNA. In RNA, cytosine, adenine, and guanine bases are used, but instead of thymine, uracil base is present. Here is a process of translation where we open up the DNA and we make a copy of RNA. And that goes out into the nucleus and is used as a template to make a protein. So the initiation of transcription occurs at a promoter region. A specific protein known as a transcription factor starts the process. The DNA strand is 3' to 5', and the RNA transcript goes in the opposite direction. Where adenine is present, uracil is substituted instead. Again, the initiation of transcription occurs at a special promoter region. The Tata box is a conserved region in adenine and thymine, the start site for transcription. It is about 50 base pairs upstream from the initiation of translation, and there's another area called the cat box, or CCAAT. The RNA is then spliced. The introns are removed so we make the entire gene, and then we splice out the introns, and exons are used to code for the protein. And this actually allows increased number of variability, so this is very important in the immune system, how they can be spliced. We could do 1, 2, 3, or 1, 3, 4, and that can result in a different protein. So again, this is called alternate introns. This is alternate splicing. So these are separate proteins. A third of all human genes have this alternate splicing that allows us to use one gene and make different gene products from it. Polyadenylation, mature beta-globin mRNA, contains about 130 base pairs of untranslated material on the 3' end, called the poly A tail. And this is believed to be important in stabilizing the RNA transcript. Pseudogenes are gene families that closely resemble known genes but don't have function. They're probably were used in the past. And there are a significant number of what we call pseudogenes. Processing, after the transcript is produced, there is a number of processes. We talked about putting on the poly A tail. But another thing we remove the introns, put on the poly A tail, and then put a methyl cap on the 5' end. And all these modifications of the RNA occur in the nucleus. Gene expression, so basically we open up a little piece of DNA, we transcribe it, we then splice out the introns, put the 5' cap and the poly A tail, and then that goes out into the cytoplasm, and this RNA is used as a template to make a protein. Translation, the completed RNA leaves the nucleus, as I said, and goes out in the cytoplasm, and in the cytoplasm, the transcript's brought to the ribosome, and the ribosome is where the translation occurs. mRNA is used as kind of a model, and then tRNAs bring in a correct amino acid, and then the ribosome goes step by step on the amino acids and builds this protein. Now, the tRNA, there's 64 possible codons with 20 amino acids, and many of the amino acids have more than one triplet. But there are certain, like AUG, codes for methionine, and that is at the beginning of every single, it's the first codon of every single protein. So that is the start codon. There's also a number of stop codons, and when we see these, the protein just breaks off. And that's where sometimes if we have a mutation, it could cause it to fall off prematurely. So translation, as I stated, also always begins with AUG. The bonding between the codon mRNA and the anticodon tRNA brings the appropriate amino acid into position in the ribosome, When the stop codon is seen, the protein is released. And then there's post-translational or post-protein things that occur. So here we have our DNA code. We then make a transcript. We then cut out all the, introns, and then this is the reading frame that makes the protein, in this case, beta globin. And this is 3.6 in the Thompson and Thompson book. Here is just another example. So each codon puts a different amino acid in the right sequence in the protein. Post-translational changes occur when we cut apart certain areas, and then they fold in the correct way. Pseudogenes, like I said, there are a number of families that closely resemble the genes but do not function. They were probably historically used. Anything that prevents the gene from being either transcribed or translated results in non-expression of the gene. Changes in genes can result in abnormal protein or lack of protein. And sometimes a very small change, such as results in a stop codon. So, self-study questions. RNA is processed to ' its final structure. Where is the poly A tail? On the 5' end of the transcript, in the intronic region of the transcript, on the 3' end, on the 5' and 3', or not all RNA is given a poly A tail. Self-study question 2. Which of a particular trait is inherited from a parent to the offspring refers to Mendel's first law, Mendel's second law, Mendel's third law, meiosis, or mitosis. And this completes the initial lecture on genetics for the genetic series.